Good day, students. In this pre-recorded video, I will discuss Module 10, which is the specific host defense mechanism. To set our expectations for this lecture session, we have the following learning outcomes. At the end of the lesson, the student can differentiate between humoral and cell-mediated immunity, distinguish between active acquired immunity and passive acquired immunity, distinguish between natural active acquired immunity and natural passive acquired immunity, artificial active acquired immunity, and artificial passive acquired immunity, and cite an example of each. Enumerate the role of the different classes of antibodies and T cells in the adaptive immunity, and briefly discuss the different types of vaccines. This will be the sequence of our discussion, so we will start with the introduction, followed by the acquired, humoral, and cell-mediated immunities, hypersensitivity and its reactions, and autoimmune diseases and immunosuppression. Let us have a quick review before we start. So in the previous lesson, we have tackled about the non-specific defense host mechanism, and this consists of the first line of defense and the second line of defense. If you can recall, the first line of defense are the physical barriers, such as the skin and mucous membranes, while the second line of defense are the mechanisms such as the inflammation, fever, activation of the complement system, and phagocytosis. Now we will discuss the specific host defense mechanism that utilizes the immune system, which is the third line of defense. Now let's start with the introduction. So as I have said earlier, the utilization of the immune system is the third line of defense against infection, and this opens the door to immunology, which is the study of the immune system and immune responses. So the word immunology came from the root word immunis, so that's I-M-M-U-N-I-S. This is a Latin word which means to make safe or exempt. So we have two primary functions of the immune system. First is to differentiate self from non-self, and second is to destroy, which is non-self. So the non-self here refers to the foreign substances or foreign microorganisms or pathogens. The immune system has two major arms. These are the humoral and the cellular immunity. For humoral immunity, this involves the antibodies, while the cellular-mediated immunity utilizes different cell types. Later on, as we go along with this lecture, we will discuss thoroughly both humoral and cellular immunity and differentiate each. Now let's proceed to the acquired immunity. We have two types of immunity, the innate and the acquired. When we say innate, this is the immunity that is present at birth and lasts a person's entire life. The innate immunity is the first response of the body's immune system to a harmful foreign substance. For example, that will be your first and second line of defense. So we have the skin and the barriers like the skin and mucous membranes. And for the second line of defense, these are your mechanisms like phagocytosis, inflammation, activation of the complement system, etc. While acquired immunity, this is an immunity that results from the active production or receipt of protective antibodies during one's lifetime. So we have two types of acquired immunity, the active acquired and the passive acquired. When we say active acquired immunity, this is where antibodies are produced within the person's body. This is through the stimulation of the immune system, either by natural infection or the administration of a vaccine. While passive acquired immunity, this is where the person receives antibodies that are produced by another source. Maybe it came from another person or an animal. For active acquired immunity, we also have two types, the natural and artificial. For natural active acquired immunity, this is achieved through infection, which then stimulates the body to produce protective antibodies against the offending pathogen or microorganism. While Artificial active acquired immunity, this is achieved through the administration of vaccines where the vaccine acts as a stimulant of the immune system to produce antibodies against the specific antigen that the vaccine represents. In line with the artificial active acquired immunity, we will discuss more about vaccines. So a vaccine is a material that can artificially induce immunity to an infectious disease. This comes from the Latin word vacca, which means cow. This is because the first vaccine was derived from the cowpox lesions to develop immunity against smallpox. It was then believed that milkmaids never got smallpox. So Edward Jenner developed a vaccine from the materials and lesions of cowpox and inoculated it to the locals. He then was first to publish the successful results of vaccination. The Chinese, also in the 11th century, used powder from smallpox scabs to immunize a person through skin application or inhalation. And Louis Pasteur developed vaccines for cholera, anthrax, and rabies, and he was the first one who used the terms vaccination and vaccine. There are certain criteria that should be met before a vaccine is released to the public. So an ideal vaccine contains enough antigenic determinants to stimulate the immune system to produce protective antibodies, contains antigenic determinants from all the strains of the pathogen that causes the disease, has few side effects, and does not cause disease in a vaccinated person. Now we will discuss the 7 types of vaccines. First, we have the attenuated vaccines, which utilizes weakened pathogens through growing the organism for many generations under various conditions or by exposing it to chemicals or radiation. So this type of vaccine should not be administered to immunosuppressed individuals since it can cause disease even in weakened conditions. So we have the following examples here. The attenuated viral vaccines such as the adenovirus, chickenpox, measles, mumps, German measles, etc. We also have attenuated bacterial vaccines such as the BCG or the Bacille Calmette-Guerin. This is a vaccine that will protect against tuberculosis infections. Then we have cholera, tularemia, etc. Second is the inactivated vaccines, where it utilizes pathogens that are inactivated through killing by heat or chemicals. So examples are inactivated viruses or viral antigens such as hepatitis A, influenza, rabies, and the Sinovac and Sinopharm COVID-19 vaccines. Third, we have the viral vector. So this type of vaccine utilizes a virus carrier that cannot cause disease such as strains of adenovirus. So we have the following examples: Janssen or Johnson & Johnson, AstraZeneca and the University of Oxford, and Sputnik V COVID-19 vaccines. Fourth, we have the subunit vaccines. So this utilizes antigenic portions of the pathogen. This is also called as the acellular vaccine. So we have the genetic approach, which utilizes DNA or mRNA. So for mRNA vaccines, we have the popular Pfizer, BioNTech, and Moderna COVID-19 vaccines. For the protein subunit, we have the Novavax COVID-19 vaccine. Fifth, we have the conjugate vaccines. So this type of vaccine is made by conjugating bacterial capsular antigens to molecules that stimulate the immune system. Since these bacterial capsules are not that antigenic, so we have the following examples: the Haemophilus influenzae type B, meningococcal meningitis, and pneumococcal pneumonia. Sixth, we have the toxoid vaccines. This utilizes exotoxins that are inactivated by heat or chemicals. This type of vaccine neutralizes exotoxins of pathogens. Examples are the tetanus toxoid and the diphtheria toxoid. And lastly, we have the autogenous vaccines. This type of vaccine is prepared from a bacteria isolated from a local infection and injected to the same person to produce more antibodies. We are now done discussing the active acquired immunity, so let us proceed to the passive acquired immunity. So we have two types, the natural and the artificial passive acquired immunity. When we say natural passive acquired immunity, this involves fetus receiving maternal antibodies. So the only antibody that can cross the placenta is the IgG or the immunoglobulin G. So later on, we will discuss each of these immunoglobulins and their function. And you also have infant receiving maternal antibodies in colostrum or breast milk. In contrast, artificial passive acquired immunity involves transferring of antibodies from an immune person to a susceptible person, for example, in plasma donation. And you also have immune serum globulin that is developed in laboratories and uses purified animal antibodies. So examples are the hepatitis B immunoglobulin, rabies immunoglobulin, and chickenpox immunoglobulin. To sum up, we have a table here for the types of acquired immunity. So when we say active acquired, we have two types, the natural, where immunity is acquired through an actual infection, and artificial, where immunity is acquired in response to vaccines. For passive acquired immunity, so we have two, the natural passive, where immunity is acquired by a fetus when it receives antibodies from the mother through the placenta, and where an infant receives antibodies through breastfeeding. And artificial passive, where immunity is acquired when a person receives antibodies in antisera or gamma globulin. Now let's proceed to the humoral immunity. As mentioned earlier, humoral immunity is one of the two arms of the immune system where it utilizes antibodies to defend the body against pathogens. Humoral immunity involves the production of antibodies by B cells in response to antigens. So what are antigens? Antigens are foreign substances in the form of microorganisms such as bacteria, viruses or parasites, or in the form of allergens such as pollen or dander, and this stimulates the production of antibodies. Since these substances stimulate the immune system, they are termed as antigenic or immunogenic. Antigens may also be in the form of proteins, large molecules of DNA or RNA, or any combination of biochemical molecules. For epitopes or antigenic sites, these are the determinants that a substance is non-self or foreign. So a bacteria is a mosaic of antigenic sites or determinants. In the case of haptens, these only act as antigen when coupled with the protein carrier. So an example would be drugs or medications. So this would also explain why there are persons allergic to a specific drug or medication since the body produces antibodies against it. There are two types of antigen processing in the body. First is the T-dependent antigens, where the T helper cells are involved in its processing, while in the T-independent antigens, the B cells are involved. So as you can see in the figure here, so this is a B cell with specific antigen receptor binds to the antigen and later on will become activated. And this activated B cell multiplies into two, which is the B memory cells and the plasma cells. So the plasma cells are responsible for secreting antibodies against the antigen. Antibodies are immunoglobulins with particular specificity for an antigen. For example, an antibody against Salmonella typhi is only specific to Salmonella typhi only and will not react with other antigen or microorganism. In addition to being found in blood, immunoglobulins are also found in lymph, tears, saliva, and colostrum. Antibodies found in the blood are called humoral or circulating antibodies. Here is a structure of a monomeric antibody. So it is Y-shaped and is composed of the Fab and the Fc region. So Fab means antigen binding fragment. So we have two, the left and the right. So for the Fab fragment or the Fab region, it contains the light chain and two antigen binding sites. For the Fc fragment or the Fc region, it contains the heavy chains. Here's the structure of the different classes of antibodies. So we have here the non-secretory IgA, IgD, IgE, and IgG, which are all monomeric in structure. While the secretory IgA is a dimer since it is composed of two monomers joined together by a J chain or a joining chain. We also have here the IgM, which is a pentamer and is composed of 5 monomers with a J chain in the middle. So we have here the immunoglobulin classes, its composition in the serum, and its features and functions. So we can have the acronym GAMDE, which represents the concentration or the order of concentration of these immunoglobulin types in the serum. So first we have IgG, which is predominant in the serum, comprises 70 to 85%. So this is the only immunoglobulin that crosses the placenta. It activates the complement and is long-lived, persisting for lifetime, and this would indicate past infection since this is involved in the secondary response. Next is the IgA, which comprises 10 to 20% in the serum. This is predominant in secretions like saliva, tears, sweat, colostrum. Next is the IgM, which comprises 10% in the serum. It has 10 antigen binding sites. Since this is a pentamer, this is formed in a primary response and will peak if we try to quantitate this in a recent infection. This will be predominant among all of the immunoglobulin classes. And this is most efficient in complement fixation. Next is the IgD, which is less than 1% in the serum and is found on the surface of B cells. And lastly, we have the IgE, which comprises less than 1% in the serum. This is produced in response to allergens and found on the surfaces of basophils and mast cells. So we have here the antigen-antibody interactions and their effects. So we have first the prevention of attachment. So the pathogen, which is coated with antibody, is prevented from attaching to a cell. Next, we have clumping of antigen. Since antibodies link antigens together, the phagocyte can easily ingest the clumped antigen. Next, we have the neutralization of toxins. So antibodies bind to toxin molecules and prevent them from damaging cells. Next, help with phagocytosis. So phagocytes can attach more easily to antigens when they are coated with an antibody. We have also activation of complement. So when complement attaches to antibody on a cell surface, a series of reactions begins that activates complement to destroy cells. And lastly is the activation of the natural killer cells, where natural killer cells respond to antibody adhering to a cell surface and attack the cell, thereby lysing the cell. And that was all for the humoral immunity. We now proceed to the cell-mediated immunity. Cell-mediated immunity is a complex system of interactions among many types of cells and cellular secretions. So it involves cells like the T helper cells, cytotoxic T cells, T regulatory cells, NK cells, and granulocytes. So we will discuss the function of each cells. So first we have the T helper cells, or the CD4. This is called CD4 since this expresses CD4 on its surface. So this cell secretes cytokines to support the humoral immune response. Then next we have the T regulatory cells, or the T suppressor cells. This will combat autoimmunity by suppressing T cells. Then we have T cytotoxic cells, or the CD8, since this expresses CD8 on its surface. So this type of cell destroy virally infected host cells, foreign cells, and tumor cells. And lastly, we have the natural killer cells, or the NK cells. This will kill target cells, including foreign cells, host cells infected with viruses or bacteria, and tumor cells. So we have here the steps in cell-mediated cytotoxic response. So first we have here a macrophage ingesting an infected cell. Then after digestion, the macrophage expresses the antigenic determinant of the infected cell and is bound to the helper T cell and the memory T cell. So the helper T cell here will later on secrete cytokines to promote mitosis and migration of the effector cells, such as the cytotoxic T cells and the natural killer T cells. So these cells will later on bind to an affected cell with the same antigenic determinant, and when it is bound, the natural killer cells and or the cytotoxic T cells will release cytokines, enzymes, and toxins, rendering the infected cell to death or lysis. Since we are done discussing the immunities, so we will now proceed to the fifth subtopic, which is the hypersensitivity and hypersensitivity reactions. Hypersensitivity, by the word itself, refers to an overly sensitive or overly reactive immune system. So we have four types, and we will elaborate each type and give examples in the next slides. So first we have here the type 1 hypersensitivity reactions. So let us first discuss the mechanism or events happening in this type of reaction. So first we have here the mast cell sensitization, where this is the exposure or the first exposure of the antigen or the allergen that causes the plasma cell to produce specific IgE antibodies which attach to the surface of the tissue mast cells and blood basophils. Second is the binding of allergen. So when there is exposure to allergen, receptor-bound IgE molecules are cross-linked by an antigen or an allergen. And third, we have here the mast cell degranulation. So the sensitized mast cells are stimulated to release granules containing histamine, leukotrienes, prostaglandins, and other potent chemical mediators, and thereby producing an allergic response. For allergic responses, an atopic or allergic person produces IgE when exposed to antigens which can cause allergies and termed as allergens. So the severity of the allergic response is dependent on the factors like the nature of antigen, the route of entry, amount of antigen, the ability to produce IgE antibodies, frequency of exposure, and length of exposure time. So we also have here the examples of allergens. So we have here animal dander such as cat dander, drugs such as penicillin, foods such as peanuts, shellfish, and dairy products, house dust, such as dust mite feces, insect venom, latex, which is present in gloves, mold spores, and pollens. Type 1 hypersensitivity reactions may be localized or systematic. When we say localized, these reactions are taking place into a specific area or organ of the body. So examples here are the hay fever, asthma, or hives. While systematic anaphylaxis, this results from the release of chemical mediators from basophils in the bloodstream. It occurs throughout the body and may lead to a fatal condition known as the anaphylactic shock. So anaphylactic shocks may happen in systemic anaphylaxis of drugs or insect venom. If this is not treated immediately, it can lead to death. With latex allergy, this is frequent in healthcare workers who always wear latex type of personal protective equipment or PPE like gloves. Latex can trigger three types of reactions. So first we have the irritant contact dermatitis. This is the common or the most common type, and this causes dry, itchy areas on the skin. Next is allergic contact dermatitis. This results from chemicals added to latex during manufacturing and causes delayed hypersensitivity. And lastly, we have the latex allergy. This is an immediate type of hypersensitivity that causes mild reactions like skin redness, hives, or itching. More severe reactions include respiratory symptoms and asthma. Anaphylactic reactions can be prevented by avoiding known allergens. So in some cases, skin tests such as scratch tests or intradermal injections of allergens are used to identify the offending antigen. So the test is considered positive if cutaneous anaphylaxis such as swelling and redness occurs at the site. So just like here shown in the picture. So this swelling and redness often referred to as the wheal and flare reaction. So once the offending antigen is identified, immunotherapy, which is hyposensitization, is done by injecting small doses of allergen several days apart to produce IgG rather than IgE. For type 2 hypersensitivity reactions, these are cytotoxic, meaning the cells here are destroyed. So a perfect example is in the case of incompatible blood transfusions, where the natural antibody destroys the incompatible blood transfused to the patient. So a typical type 2 hypersensitivity reaction shall follow the steps. So first we have a particular antigen binds to the surface of the cell or the target cell. Then an antibody will then bind to the antigen. Then this will initiate the binding of the natural killer cell to the antibody. Then the natural killer cell will then release cytokines that promotes cell lysis. For type 3 hypersensitivity reactions, this involves immune complex reactions. So when the body is infected with a bacterial pathogen, the body tends to produce antibodies against the bacteria, and that will result to an antigen-antibody complex. So these complexes will later on be deposited into the organs, causing hypersensitivity. So for example, in rheumatoid arthritis, which is an autoimmune disease, this produces rheumatoid factor, which is an antibody directed against the Fc region of the IgG. So when immune complexes forms, it will be deposited to the joints, thereby causing inflammation. So other examples are the systemic lupus erythematosus and Streptococcus pyogenes infections, where immune complexes are deposited to the heart, causing rheumatic heart disease, and into the kidneys, causing glomerulonephritis. So with type 4 hypersensitivity reactions, so these reactions are referred to as the delayed type hypersensitivity or the cell-mediated immune reactions and are part of the cell-mediated immunity. So this type of hypersensitivity will involve the antigen-presenting cell. So usually these are the Langerhans or the dendritic cell. So this antigen-presenting cell will bind to the antigen and will bring that into the nearby lymph node and presenting it to the T helper cell. So the T helper cell will transform to its subtypes, the T helper 1 and the T helper 17 cell. So these cells will release mediators to attract effector cells such as the cytotoxic T cell and the natural killer cell. So a perfect example also of this type of reaction is the Mantoux skin test or the tuberculosis skin test. So this is where tuberculin, which is a purified protein derivative prepared from substances from the bacteria that causes TB, is injected intradermally. Then after 24 to 48 hours, redness and swelling would be present in a positive reaction. So to sum up, we have here a table for the types of hypersensitivity reactions. So we can classify the 4 reactions into 2. So we have the immediate type hypersensitivity. So under this type is the type 1, which is the anaphylactic reaction, type 2, which is the cytotoxic reactions, and type 3, which involves immune complex reactions. For delayed type hypersensitivity reactions, we only have the type 4 hypersensitivity reaction, and this involves the cell-mediated reactions. For the last subtopic, we will discuss briefly the autoimmune diseases and immunosuppression. Autoimmune diseases. So autoimmune diseases results when a person's immune system no longer recognizes certain body tissues as self and attempts to destroy those tissues as if they were non-self or foreign. So these are also the results of type 1, 2, 3, or 4 hypersensitivity reactions. So for type 2, we have myasthenia gravis as an example. So myasthenia gravis is an autoimmune disease where the body produces antibodies against nicotinic acetylcholine postsynaptic receptors at the neuromuscular junction of the skeletal muscles. So this causes skeletal muscle weakness and rapid muscle fatigue. So for type 3 hypersensitivity reactions, we have here the examples rheumatoid arthritis and systemic lupus erythematosus, which is a systemic autoimmune disease that commonly affects the skin, joints, and kidneys. Immunosuppression. So the terms immunosuppressed, immunodepressed, and immunocompromised all refer to a dysfunctional immune system. So this may be inherited or acquired. For acquired immunosuppression, an example is the acquired immune deficiency syndrome or AIDS, which is caused by the virus human immunodeficiency virus or HIV. So the HIV decreases T helper cells, which is vital for the production of antibodies. Other causes are exposure to chemotherapeutic drugs and irradiation. While inherited immune suppression, this is a result of deficiencies in antibody production, complement activity, phagocytic function, or NK cell function. So examples here are the severe combined immunodeficiency or the SCID, where patients are deficient of B cells or T cells or both, and the George syndrome, where there is absence of thymus and parathyroid glands during birth. So this will have a deficient number of T cells since the site of maturation is not present. That's all for our discussion about the specific host defense mechanisms. I hope you have learned a lot from this lecture. Thank you and have a great day.